Here is the answer. You check your own answers, okay? Who would like to answer? You may unmute. Nobody would like to answer. I will give you the answer na lang. And then you determine your answer if it is correct. Okay, and then if it is wrong, then you, what do you call this? You try to solve it by yourself, okay, until you can perfect it. This one, this is the answer. Did you get it? Correct? I-try ninyo sa ano. Be honest enough to accept if you are wrong. And then you try to correct your solution. Because there is a tendency that this calculation will come out in the exam. So better, you have to know how to solve this. For example, this one, your glucose. So how to arrange is A, B, C, D. Okay? So your A, glucose is 20, urea 30, sodium chloride is 150, calcium chloride is 10. So 380 milliosmoles. The tonicity, A, B, C, D. Your urea is penetrating. So this will not affect. This one is a milliosmol, okay? B, C, D. D is hypotonic. Isotonic, hypotonic, hypertonic, and hypotonic. How did you get this? That one. G times C. Diba? Yan ang formula. So, glucose is 20. Urea is 20. Sodium chloride. Ay, ano ba yan? Glucose is 30. Okay. 30. Urea is 10. Sodium chloride is 60. Oh, for 60. You arrange it from the highest to the lowest, from the lowest to the highest. Okay? Sige na. Basta you try to check that. Okay? We will go to endocytosis. A region of the cell membrane invaginates to form a pocket. If you are not sure of your answer, then if somebody in the class knows, then you teach your classmates. Endocytosis. A region of the cell membrane invaginates to form a pocket containing extracellular fluid. So the pocket then pinches off to produce intracellular membrane-bound vesicle. So it will get inside, whereas the exocytosis is the opposite. Okay? The membrane-bound vesicle, the vesicle will fuse with the plasma membrane and then extrude its contents towards the outside, towards the extracellular fluid. The opposite is endocytosis. Endocytosis, you form a pocket, so sort of pinch off and then bring the membrane, the vesicle inside the membrane, the cell. Types of endocytosis, fluid. This is also the pinocytosis. The solutes and water are non-specifically brought into the cell from the extracellular fluid via the endocytotic vesicle. This one here. Non-specific uptake of solutes and water. So this will form a vesicle and then brought inside. This is fluid endocytosis or pinocytosis. Phagocytosis is specialized cells form extensions of the cell membrane called pseudopods, pseudopodia. And then this engulf, bacteria, for example, or other large objects such as cell debris. The vesicles that form fuse with the lysosomes, which contain enzymes and other molecules that destroy the vesicle content. Then the bacteria will be acted upon by the lysosomal enzymes. And so this will die, okay? So especially if your immune system is good, then the bacteria can really die because you have lots of lysosomal enzymes. Receptor-mediated endocytosis. In this type, the cell recognizes a specific extracellular ligand or a chemical that binds to a receptor. It could be a hormone or chemical that binds to a receptor in the membrane. In the membrane, there is a receptor in the membrane, and it is clathrin coated because it has receptors. So this will form a pit, no? And the ligand will bind to the receptor, in the pocket. And then the vesicle is brought inside. The proteins will be released from the vesicle. So there is the unbound ligands, and this will be acted upon by the lysosomal enzymes. And some will find its way into the nucleus. The receptors will be recycled. This will go back to the cell wall. So nature really recycles. If nature recycles, we have to recycle. Our grocery bags, we have to recycle. So, yun, the receptors will be recycled. The sodium-potassium pump is a primary transport, okay? Transport. It is a transport process that pumps sodium ions outward through the cell membrane of all cells. At the same time, pump potassium ions from the outside to the inside. This is responsible for maintaining the sodium and potassium concentration difference across the cell membrane. This maintains the negative electrical voltage inside the cells, and this controls the volume of the cell. This establishes the ionic concentration that helps drive the co-transport of another molecule. This one is electrogenic in its activity. Why? Because it transports 3 sodium out for every potassium that will go in, keeping the interior of the cell negative relative to the exterior. You notice that sodium is positive, right? So 3 sodium going out and 2 potassium going in. That is the leakiness of potassium. But you notice that only 2 potassium is returned back to the inside, while 3 sodium is pushed out, returned to the outside, right? Remember, you have more sodium outside and you have more potassium inside. But since it's only 2 potassium, so there is 1 lacking. So there is negativity. There is 1 negative na because of the 3 sodium and only 2 potassium, so it's not balanced. So inside is negative relative to the outside because sodium is 3 outside and only 2 potassium inside. So kulang siya ng positive, lacking positive. And this involves energy, sodium potassium ATPase enzyme. And so this involves energy expenditure, this pump. No? So with the action of your ATPase enzyme, 1 ATP is used for every exchange. And the ATP will be reduced to your ADP, okay, plus 1 phosphate. The phosphate will be utilized as energy in the process of pumping the sodium out and potassium in through the use of your sodium potassium ATPase.